Hello Year 12, it's me again with another kind of recorded lesson for you. It's quite a short one, this one. I'll also send you a link to another video clip that I'd like you to watch. First thing, a bit of recall, focus on the first letter. So just pause the clip now. Can you jot down what each letter or each word in this particular sequence relates to? Okay, hopefully you've managed to realize to do with classification, and you should have come up with the following sequence. All being well, you should have come up with domain, kingdom, phylum, class, order, family, genus, and species, all to do with classification, looking at similarities between organisms. Well, a lot of the work that I've been covering with you in the past few weeks has been to do with DNA, protein, transcription, translation, mutations, differences between amino acid sequences, differences between DNA sequences. So we're going to look at this idea of looking at classification, where we're looking at similarities between DNA and protein. So the title of today's work, if you like, is classification using DNA and protein. I'm just going to have a quick look at this particular part first of all. And what you can see along here is this sort of idea of a phylogenetic tree, and we're looking across the top. We've got human, macaque, dog, bird, frog, and a lamprey. And what this little image represents is a chart to show the differences between amino acid sequences and differences in the sequences of a particular protein. In this case, we're looking at one of the protein chains in hemoglobin, and we're looking at the number of amino acids that are different between the hemoglobin protein in human compared to macaque. So there are 8 amino acids different between a human and a macaque. Human compared to dog, we're looking at 32 amino acids different. So both macaques and dogs have hemoglobin, but the structure of their hemoglobin is slightly different to human. If we look at human compared to bird, in this case it looks like it's a chicken, we're looking at 45 amino acids different. Down as far as the frog, yes, they've got hemoglobin too, 67 amino acids different to human. Right the way further down at the end down here we've got a lamprey, something described as quite a primitive fish. This has got 125 amino acid differences. So in terms of classification and looking at similarities, we don't just have to look at physical appearance of the organism. We can look down at more fundamental and start looking at the protein itself and the amino acid sequence. So providing you know the amino acid sequence, you can compare using the same protein. It's important it is the same protein. You can compare differences between the protein sequences. In this case, again, still looking at hemoglobin, that same kind of idea. So we've got 146 amino acids in total in this particular hemoglobin chain, because it's a quaternary structure protein, so there's more than one kind of primary sequence involved. And so human, obviously human don't have the same hemoglobin amino acid sequence that makes it up. Compared to gorillas have one amino acid that is different compared to human hemoglobin. The rhesus monkey, similar to the macaque from earlier, a difference of 8. The mouse hemoglobin compared to humans got 27 differences. There's that chicken again, there's the frog again. And so you can just kind of see you've got this idea of there being some differences between the amino acid sequences in different organisms. So it's a useful way of looking at classification by specifically looking at the sequences of amino acids in a given protein. Now, because we know how the amino acid sequence is coded for, we know the genetic code, and this idea of a triplet coding for a particular amino acid, we could take that one step further back, if you like, and look at DNA sequences. It's the DNA that codes for a particular amino acid and hence gives us that particular protein structure. So we could look at the DNA sequence rather than looking at the amino acid sequence. So this particular slide, got a little bit too far, not the rabbit yet. This particular slide looks at a very short sequence of DNA, and so we have an unknown species at the top, which has the sequence ACT, GCA, GCC, and we've got 4 species. Make a look just how similar is the unknown species DNA sequence to the 4 unknown. So if we start with ACT, that first triplet, you can see there are 2 down here, species 2 and species 4, that have a similar first triplet. And we look at the next set, and then got GCA. Species 2 has got GCT, species 4 has also got GCA, so The unknown is very similar so far to species 4, and our unknown has GCC, and finally the last one has GCG. So there's only one base that is different with our unknown species compared to species 4. So it suggests perhaps that the unknown species is most closely related to species 4. They've got the most similar DNA sequence. So just kind of two methods where we can look at either the amino acid sequence and look for similarities. Those that have the most similar amino acid sequences are most likely to be closely related. Those that have differences further apart are more likely to be more distantly related. So it's an indication of kind of a classification and an evolutionary timeline, if you like. There's another example. If we kind of go back to the immunology topic that we studied earlier this year, we can also use immunology to come up with an idea of similarities. That's what the little rabbit picture was for. And this is just a technique that isn't used as much now. It certainly was used quite a few years ago to kind of compare sequences. And it does work reasonably well. The principle is as follows. The rabbit is just the organism that's going to be used to help generate some antibodies. So what we would do is we'd take our rabbit and we would inject the rabbit with a particular protein that we want to compare across a number of species. So in this case, we're going to take a human protein, hemoglobin. We're looking at hemoglobin earlier, so we'll kind of continue with that thread. So we're going to take human protein, human hemoglobin, in this case, just shorten that to HB, and we're going to inject some human hemoglobin into our rabbit. Okay, so a pure rabbit's going to get some hemoglobin injected into it, and as a consequence to that, because the human hemoglobin injected into the rabbit, there would be an immune response and would produce B cells, which would result in production of antibodies, and so we get an increase in antibodies within the blood of the rabbit over a period of weeks following the injection. So with these antibodies, they would be specific to human hemoglobin. So all these antibodies wouldn't be the same. Human hemoglobin is quite a complex shaped protein, so there'd be a number of different antibodies produced that would target particular sites on the human hemoglobin, but we'd get quite a large number of antibodies generated in our rabbit. And it's the antibodies from the rabbit that we're now interested in. That we're going to use those to look at how similar the human hemoglobin is compared to other organisms. So we've now got our antibodies from our rabbit. So what we're going to do with those is we're going to isolate and remove some blood from our rabbit and remove the what's called the serum. This is the plasma component, and that plasma part would contain our antibodies. So we can use something as simple as centrifugation to sort of spin our blood sample from our rabbit. We'll get our red blood cells, and white blood cells, and platelets, and we're left with our plasma. We can then remove the plasma, and in that plasma we would have the antibodies that were generated in response to the injection of the hemoglobin. Next step, if we were to take some of this plasma, which has the antibodies in it, and we were to mix it with hemoglobin from a number of different species, we would get what's known as a precipitate. There'd be an agglutination. The antibodies would bind to some of the hemoglobin if they have a complementary shape to the binding site on the antibody. So we'd get a degree of agglutination and we would form some sort of precipitate within our sample. If we mixed it with human hemoglobin, we'd get the maximum amount of precipitate because it was the human hemoglobin that resulted in the antibody production in the first place, so we expect a really good match. But if we were comparing and mixing these antibodies from the rabbit with another organism's hemoglobin, we wouldn't quite get the same match because the hemoglobin would likely have a slightly different structure, because it would have a different primary sequence of amino acids, because it's likely to have a different DNA sequence to begin with that resulted in its production. So we can then compare the amount of precipitate that's formed to give us an indication of how similar the amino acid sequences were between these particular proteins. So for example, we would end up with kind of a bit of a range. The greater amount of precipitate would be where we've got the greatest similarity. The smaller amount of precipitate would be where we've got the least similarity. So it may be that if you look at this kind of example down this bottom corner, I'll just zoom in for a second. So down here, just an example of a precipitate. It's not quite the one from this experiment, but we've got a lot of precipitate at this end. This might be our kind of comparative, where it's human hemoglobin mixed with the antibody from the rabbit, so we get the maximum amount of precipitate. The next sample might be from some sort of closely related species, whether it's a gorilla or a chimpanzee or an orangutan. We get quite a lot of precipitate because the hemoglobin is very similar in terms of its structure due to its the amino acid sequence to human hemoglobin. And then we might have another organism, so we wouldn't get quite as much. And further down the line, we'd end up with some organisms hardly producing any precipitate at all because the hemoglobin from that particular organism is so dissimilar in terms of its tertiary structure that the antibodies from the rabbit just wouldn't bind to it. So it's a useful procedure where we can use kind of immunology to ascertain how closely related organisms are. So it was a technique that was used quite a lot a few years ago. The main technique that's used these days is looking at the actual amino acid sequence itself, if you can determine that, or even better still, going one step further and looking at the DNA sequence and comparing those. So the next step that you're going to do is actually to look at trying to do that DNA comparison. Now the example that I showed you a few seconds ago, if I go back to that one, just looked at 9 single bases, so 3 triplets, so a really short sequence. In reality, the DNA coding is much longer than that. So this wouldn't be something that you could do easily by eye. This is where computers really come into their own, and there are huge databases set up used by scientists right throughout the world to compare particular DNA sequences, and you're going to have a go at doing that. Okay, so the information you want is at the bottom. It's another YouTube clip that I'd like you to go to. This is a set of YouTube video clips that I highly recommend that you look at. They're done by a gentleman called Paul Anderson. He's an American teacher who's done sort of online learning for many years. I use quite a few of his clips and lessons, so it's certainly worthwhile having a look. He'll cover physics, chemistry, biology, lots of different topics, but he's going to kind of talk you through how to do a particular analysis. So the details for the clip are at the bottom, but I'll also send you through the link, and I want you to follow the steps that he suggests for doing a comparison between the DNA sequence from a particular protein from human of your choice and seeing how closely related it is to proteins for other organisms. Now the instructions that Paul Anderson gives you in the clip, he made this video a few years ago now, and the website they refer to has had a few updates since then, but you can still follow along and still do the same analysis. So what I want you to do is to watch the clip from Paul Anderson. That's part of the Bozeman Science Suite on YouTube, so you can have a look for that and have a look, see what he does, and I'd like you to send me an image back of the final comparative phylogenetic tree that you can generate. So it shows you how to do that. It says you can choose slanted, and you'll know what that means when you watch this clip. You can choose slanted. When you come to use the website, you need to go to the Tools option over towards the right hand side of the screen, and you can choose slanted from in the Tools option, and then you can get a screen grab and email me the screen grab of whatever you've managed to do. Okay, so I look forward to seeing your examples, see how well you've done. Choose a protein that you know the name of, so you could choose something just as simple as hemoglobin. There are different chains. It's often using the American spelling of hemoglobin, so H-E-M-O to start with rather than H-A-E-M that we would usually use. So have a look for something like hemoglobin, or actin like he did, or myosin, or another protein that you may know the name of. Okay, look up some of the proteins maybe and see if you can do a comparison. Okay, good luck, and I look forward to seeing your clip.